Yep, so this is where we left off, low road, high road. Here's just one more view of the whole thing. So I think this is the example that's often used. In fact, it is the example given in that chapter you read is you're walking in the woods, you see something that looks like it could be a snake. Maybe it's a snake, maybe it's a twig. Yeah, that's a snake. So it comes in, you have a slow path, that's the low road. Where are we at here? It goes through the eyes. Here we are in the thalamus. And we have this fast path. It slips right down here into the amygdala where it recognizes it as a potential threat. But the amygdala doesn't really do much in the way of visual processing. Just that it's coming in here. You're getting sort of a preliminary vision, a view of what it might be. Probably tapped into memories as well. And that's going to produce the initial fear response. So, hey, okay, I should stop. Goes to the visual cortex, and like, yeah, that's a snake, no doubt about that. And then it comes down here and reinforces the response, so you continue to stop and maybe back up. Okay. All right. So now let's look at the amygdala in more detail. And here, there's that sensory cortex and sensory thalamus feed in, comes into the lateral nucleus. Here they have the lateral going into the basal. For the most part, this complex in and of itself is called the basolateral complex, and in a few slides that comes up as well. So this is mostly input. Here, this is also considered input. We don't really talk too much about the accessory basal nucleus, and we're not going to talk too much about it right now. And the central nucleus is generally believed to be an output region. Okay. There's also the medial nucleus of the amygdala, and we're going to talk about this one when we get to fear of predation. This is where the smell information comes in. Both the main and accessory olfactory bulb has a direct line into the medial nucleus. So the lateral nucleus receives stuff primarily from vision, auditory. I think that's probably it. Not too much somatosensory. But mostly vision and hearing. So what you see, what you hear, and this is more what you smell. For us, we don't really use that as much for fear, but for other animals, yes, they do. Okay, so we could also think of these things as being broken up into both a striatal division, that's the central and the medial nucleus, and a cortical division, which is more the lateral and the basal. And this is based on the kind of neurotransmitters that these particular cells contain. So the central and the medial amygdala contain primarily GABA. That's the main inhibitory neurotransmitter, and this is very comparable to the striatum or the basal ganglia. If you remember, maybe, when I was talking about dopamine basics, I did a little side trip into basal ganglia motor control, and I said anything that's in the basal ganglia, caudate nucleus, globus pallidus, they contain GABA. They're inhibitory. And anything coming in from the cortex, anything coming in from the thalamus, tends to be more glutamate. It's excitatory. And that's the same pattern we see here. So this is looking at a particular enzyme that makes GABA, glutamic acid decarboxylase. And in this part of the amygdala, that is the central amygdala and the medial amygdala, in the rat amygdala, you see a lot of this, okay? This is the caudate putamen, and notice it's sort of an extension into the amygdala. So it's not all that different in terms of the neurotransmitter that it makes. We look at the lateral amygdala over here. That's the lateral, basolateral. This is, I believe, the accessory. These are all kind of an extension of the cortex, and these are primarily glutamate. Okay, so central and medial tend to be more GABA inhibitory and pretty much an extension of the caudate nucleus, whereas the lateral, basolateral, that complex is more a extension of the cortex and tends to be glutamate. Okay. It has far-reaching influences. So here is the basolateral. This is the central. Now this will be coming out of the central nucleus, and there's a bunch. I don't want you to memorize all these. Obviously, I don't want you to memorize any of this stuff. Just know it. Be able to have a general idea of it. We see that the central nucleus sends input or output to dopamine, norepinephrine, acetylcholine area, so ventral tegmental area, locus coeruleus, the lateral dorsolateral tegmental nucleus. So what we're seeing here is arousal, we're seeing increased vigilance, and we're seeing cortical activation. So when you're put into a fearful situation, you want to have your brain aroused, vigilant, make sure it's prepared to take in important information, and also activated. This is the periaqueductal gray. So in terms of rats, what you're going to get is this freezing response. But if they can get away, it's also going to produce the running, the flight response as well. And then finally, the paraventricular nucleus, we see ACTH, we see glucocorticoids. These are the ones that we talked about in the developing brain. Okay? So all of these are getting information then from the central amygdala. So what you get is a coordinated response. You get behavioral response, you get hormonal response, and you're also going to get that conscious awareness in the cortex. Okay. So now let's look at Ledoux. Joseph Ledoux from NYU. If this is something that really like, oh, this is really cool. He's got a bunch of books that he's written over the last 10, 15 years. And not just on fear, but on emotions in general. And yeah, they're pretty good. They're sort of this in-between, not really a textbook, not really for a neuroscientist, but good for somebody who just wants to learn more about how the brain works. So anyway, Joe Ledoux, he also has a band. They have a band called the Amygdaloids. I'm not sure if they still do this or not, but yeah, it's a bunch of neuroscientists. They got together. They play in New York City. Okay, so in any event, what we're going to do, I'm going to introduce you to this experiment just for a number of reasons. One is to work through some of this circuitry, and the other thing is to look at this technique, which we already talked about earlier, in terms of using this drug called muscimol, which is a GABA agonist, to temporarily inactivate a brain area. All right, so what is this study about? First of all, the prevailing model of conditioned fear is what I just went over. The lateral and the basal nucleus are the inputs. That's up here. And the central nucleus, we tend to think of it primarily as the output of the amygdala, but its role is really not as clear as we would like to think. And the inputs to the central amygdala, they point out, are very similar to those in the lateral and the basal nucleus. So, in other words, this is receiving the same inputs as these two areas are. So if that's the case, this may be more than just a output. Okay, it may also be involved in the input of this information. So they wanted to re-examine the role of the central amygdala in conditioned fear. They're going to use temporary inactivation with muscimol, and we're also going to do is look at a study where they block protein synthesis, which would be necessary for making new memories in the amygdala. And what this is showing is that they're going to either inject here or infuse here and see what happens. So, temporary inactivation. We already talked about this, results from this, but this is how it works. Here's the GABA receptor complex. When GABA binds, the ion channel opens up, chloride comes through, and inhibits the cell. So by putting muscimol here, it acts on the GABA site and results in temporary inactivation of that particular nuclear group, that particular brain area. And so they can do it here, or they can do it here. Okay, so that's the basic design, and we'll pause here and come back to talk about how they did it.